Welcome, everybody. This is the May 13th meeting of the IMAG MSM Working Group on Multiscale Modeling and Viral Pandemics. Today, our format is a little bit different. We have Fred Adler talking about his work with the viral evolution subgroup, and John Rice talking about the dissemination outreach subgroup and some of the things they've been doing, and then also the planning for the big IMAG MSM virtual conference later this year. As always, the meeting is recorded. It's being streamed, and it will be available publicly later for people who are unable to attend. I'm really pleased to see that we have so many people coming for essentially a more business-oriented meeting, although I think we'll learn a lot about what's been going on from that. The usual slide introducing myself, Reinhardt, Bruce, and Jim, and our email addresses for your comments and suggestions. Our Slack and other sources of information. I think these are particularly important to think about in the context of both the dissemination outreach components of the working group activities and also meeting organization, and potentially for the dissemination of the work that Fred has been doing. And the next thing is to call for any short announcements for this week. Is there anything somebody would like to bring up quickly before we move on? No? All right. The usual reminder that if there is any thing that we should know about how this working group has helped you out, that would be great. We are trying to document what the working group is doing so that we can make an argument that we're useful. And so any help that you can provide in that area will be appreciated. Next week, we have a single speaker, Rusty Irving from GE, one of the developers of the digital twin ideas. And of course, we've talked a lot in various ways about digital twin concepts. Last week, I think in particular, we heard some very digital twinny models. That will be interesting. May 27th, we have Philip Ball, a noted science writer, and a talk on thrombosis. And you can see the list going forward. We have quite a few interesting talks coming up. Starting around mid-June, we have plenty of empty slots. And so if you would like to speak or you have suggestions for speakers, please do let us know and we'll do our best to get that onto the agenda. The rules of the meeting don't really apply today because we're not having a meeting of that kind. What I will say is that we will, as I mentioned, switch the order. So instead of our traditional seminar style, where we have both seminars and the questions afterwards, people should feel free to, Fred, is it okay if people comment during your talk, or do you want to give the talk first and then take comments?
No, definitely, please interrupt.
Okay, in that case, the gloves are off pretty much. I will try to keep things on time so that both, actually, is John on the call? I don't see John on the call yet. So I
haven't seen him on yet.
Okay, so until John shows up, Fred, you have the floor.
Okay, thank you. Okay, does that look okay? Good.
Looks
so, great. Thanks. So this is the report from the viral evolution subgroup. So I signed up to give this talk like a month and a half ago in order to give us a deadline, which worked because we actually have gotten together a few times and put this together. So I kind of like to think of this evolution topic in terms of Daniel Dennett's somewhat annoying book, Darwin's Dangerous Idea, where he talks about evolution being a universal acid that sort of can break everything down. And I see that thinking evolutionarily, that is what's sort of going on under the hood with this virus, breaks down a lot of sort of some of the goals of this group, and I think in a good way to challenge them. So here's the sort of the overview of the topics. I'm only going to say a very brief word about each of these that I think evolution touches on. So the pandemic begins with some sort of host switch, which often involves an evolutionary step. Then we've seen a tremendous amount of diversification, which definitely is evolution in the sense of increasing genetic diversity. And then there's various forms, this diagram isn't totally coherent, but there's genetic drift, which is diversification that doesn't have direct selective consequences. There's evolution that occurs through interactions with a whole bunch of other stuff, including the host and other pathogens. There's selection itself, and then this sort of dot is leading into where we show up as intelligent agents trying to deal with this with various control mechanisms such as treatment. And then, of course, the evolution of virulence, or how deadly the virus itself is. Okay, so the plan for this is just to sort of mention some issues related to each of these things. And so first is diversification. And of course, identifying the viral source has been challenging. And this is an issue that shows up throughout the evolutionary modeling component, which is we have very incomplete data. We don't necessarily have the bat or the pangolin who was carrying this virus in the first place. And reconstructing phylogenetic histories has to deal with these many unknowns. Population structures, mutation rates are high. They mess up the genome in various ways that are not necessarily handled by standard phylogenetic methods. Recombination, which occurs commonly in some viruses, not so commonly in coronaviruses, as far as I know, can really throw off our ability to reconstruct the past. And it's certainly a personal frustration for me is that we very rarely can link phylogenetic data or genetic data with patient data to know who got a bad case and, even how old they were. And so, I think multi-scale modeling has really important roles in thinking more mechanistically about reconstructing the past of this virus to include things like population structure and different forms of selection, and perhaps having a role doing some optimization of those methods. Okay, so this is sort of the evolution sort of setting the stage for the kind of viral diversity we deal with and using that to reconstruct the past. And here's just one example of a paper. I was looking at the UK, and I didn't want to emphasize any of the key points, but there's some issues here they're really sort of non-standard for a lot of phylogenetic reconstruction. There's all these unsampled lineages, which are dotted lines are all over the places. There's importation of cases that start new lineages, and then there are all these lags built into the system before you see something, just based on either symptoms or just happening not to run into that case. So many of the issues that people deal with in phylogenies, if you're trying to build a phylogeny of birds, you don't have to worry about this kind of stuff because the timescales are much different. Okay, so then this one, in a way, is at the heart of what this whole group is about, in my view, interactions. And so, evolution, if it's by selection, occurs through interacting with stuff. And what I've done here at the bottom is I just cut and pasted the names of a bunch of the other subgroups, all of which are affected potentially by evolution, because those are the interactions that drive selection on this virus. So what hosts look like, right? Their genetics matters, right? Viruses enter cells through biochemistry, and at least for other viruses like HIV, we know that biochemistry matters. Immune responses differ between people because the immune system is famously genetically diverse among people. That matters. Everybody's going to be different. And then the immune system itself evolves through things like affinity maturation, that is making antibodies that are more and more supposedly effective. And so it evolves on a time scale comparable to or faster than that of the virus. And of course, interactions with other pathogens, and one thinks particularly here of the seasonal coronaviruses, depends on their genetics, and those genetics interact with the evolution of the hosts in complicated ways. So here's just 5 of the subgroups that I had room to put on here that are affected by viral evolution. The immune response, like I've talked about, transport, mode of entry are all affected by both the biochemistry and evolution of the virus and that of the host. Replication and release is really the flip side of that. It's another piece of how the biochemistry of the two organisms interact. modeling individual responses to treatment will depend not only on the treatment, but on the genotype of the virus and of the host. And of course, virus-virus interactions are going to be highly sensitive to the genotypes of both of the players. Drift, I think, is particularly interesting. So, on the face of it, drift, which means just generation of genetic variation that has no selective impact, is our friend, because it's what gives us the information to be able to reconstruct the history of the virus. But it also can lead to challenges for things like testing. So in fact, yesterday I was talking to the people at BioFire here who make tests, and they have assured me that of the 1 million variants out there, their test gets every single one of them. And I actually believe them. And so it's quite a challenge to make tests that have a broad purview like that, but do not, say, detect other coronaviruses accidentally. I think there's some really interesting modeling issues, things that I've never dealt with myself at all, but thinking about how one actually would optimize PCR or antigen testing to deal with this variation, or thinking about sort of more biochemical models that can take sequence, actually compute structures, and then sort of computationally test whether different protocols will detect those viruses. And here's a paper that's not about coronaviruses by Kennedy and Dwyer. This is actually in a system in moths of some sort. And I just wanted to emphasize that the drift is very much a function of the population structure, and population structure really is a function of transmission itself. So if my initial infection is one virus that starts to grow, that has very different effects than if I have 30 viruses in terms of the strength of drift. So drift strength is very much linked to the actual epidemiology. Selection tends to be what we think of as being at the core of evolution. And so viruses can face a whole bunch of competing selection pressures. Transmission between hosts is a very different thing from evolution within hosts. Evolution within hosts depends on other viruses that may or may not be around, but certainly most primarily on interactions of the immune system. Multi-scale modeling tries to interface those internal dynamics, what's going on within hosts, with what's going on outside hosts. And once again, using some kind of sequence to structure calculations might help us in order to be able to predict how selection, in fact, will operate in different environments within hosts. Virulence is, a central concern. What is going to happen to this thing over the long run? There's this general idea out there, which I think is often the case, that viruses become less virulent. But there's multiple reasons for that, and there's the complexity of modeling virulence is really quite high. Host heterogeneity plays, I think, a very large and sometimes underestimated role, in that viruses, from their perspective, Jim and I are very, very different biochemical environments, and it's hard for them to evolve to be able to deal with all of those things. And so host heterogeneity matters and affects the evolution of virulence. Treatment itself might promote virulence. If everybody's treated, then the virus might get more nasty. The same idea has been promoted for vaccination, even though I'm fairly skeptical of that. Immunity has quite interesting effects on virulence. That is, if individuals mount a stronger immune response, the virus may act to be either more or less virulent in response to that. And here's an example. This is an archive paper, so we don't necessarily have to believe the results, but this is this classic idea in the evolution of virulence, that there might be a trade-off between transmission and virulence. That is, viruses that are better at transmitting have to build up to higher titers, and therefore are going to be more virulent, make people more sick. Again, that's often not necessarily the case. And they put some letters and made some graphs here, which I'm not going to attempt to explain, but it's basically they're arguing that there is evidence for such a trade-off with SARS-CoV-2. And I did not have a chance to study that to see how compelling that is. Finally, on the control side, and this is, of course, the universal acid thing, is in principle, evolution could erode everything we try, right? It could make transmission higher. We've seen examples of that with the British variant. we could see treatment escape, and that was a concern even back in the Regeneron days, which is, I think, why they used 2 antibodies instead of 1, because you can basically compute that it'll be fairly easy for the virus to evade a single antibody. Modeling how imperfect vaccines can be invaded. Obviously, vaccination has changed the selective landscape vastly, and there is some selection to evade vaccines. And finally, how developing immunity, that is, if we're all hanging around waiting for the second wave of this virus, is it going to have to deal with that just as influenza does and change itself in some way, either immunologically or in terms of how sick it makes us? Here's just an example of evolutionary escape from neutralizing antibodies that people are worried about. And these guys were comparing sort of different scenarios. If there was 1 mutation, 2 mutations, 3 mutations, and 4 mutations, how many of the population, how many members of the population will we see getting infected by these different types? And once again, I'm not going to attempt to go into detail. So that's it. So that's my grand overview of sort of how I think viral evolution sort of is related to pretty much everything we're talking about. And here's just a quick summary. So back in the early days of the white papers, Reinhardt sort of laid out a framework, which I thought was really good, of thinking about sort of this topic in terms of 4 questions, right? What can't we do at this time? I would say right now we can't detect the detailed course of evolution very well, if at all, even statistically. And that's just in the nature of the beast. There's a huge amount of stochasticity in there at many levels, mutation, drift. And so I think we just have to be realistic about accepting that. I don't think we really understand trade-offs between levels of selection. That is, if are mutations that make it more transmissible also likely to make it more or less virulent? We don't know that. That's actually extremely difficult to show empirically from the sort of data we have. And I think we really have a lot of problems dealing with heterogeneity among people, that people are really different, not just genetically and immunologically, but also just in terms of overall health status, diet, and all these other things. That heterogeneity may matter. We can't address all of that. I'm taking a quick look at the chat here, if you'll excuse me. Right. Yeah, I agree. We can somewhat predict the future, but not very well. Yes. I agree. Yeah, thanks. I'll never use the words predict and forecast correctly. So you can predict that I'll use them wrong. No, forecast that I'll use them wrong. I'm sorry. So, so then the second question Reinhardt laid out is what capabilities we lack. So I think one thing that is to me really exciting is thinking about coupling in sort of in real time, mathematical modeling, informatics of the sort that I was talking about, like reconstructing phylogenies, and ideally some experiments, bringing different strains into the lab and doing some basic fitness testing to try to map fitness and phenotypic landscape of pathogens as new types emerge. I think that is actually realistic. I think we lack this capability, as I've said, to measure selection pressure very effectively. And, even knowing population sizes, we had that great talk a few weeks ago about how much do they all weigh. That's very fun, but in some ways what we really care about is the effective population size from the genetic side, and I don't think we have the capability to estimate that very well because we have to know infectious doses. I don't think, I've tried forecast to see if that's the right word, I don't think we can forecast the fate of new mutants particularly well. So I had some good conversations with some people about the D614G mutation that's been hanging around, or maybe it goes the other way around. And they're still debatable whether or not that spread due to selection or due to drift. And the movement of people is very complicated and difficult to predict. And then finally, this idea that's been thrown around, which I also really like, of model ensemble averaging. So can we actually develop a set of models for these evolutionary scenarios that have some statistical predictability and maybe use real-time data and some sort of ensemble averaging to give us a sense of how predictable the future is. So then Reinhardt challenged us to actually do something. How can we develop those capabilities? I don't know the answer, and I'm hoping others have more specific suggestions. There's been a lot of nice work done on certain model systems. I mentioned influenza, HIV, hepatitis C, and vesicular stomatitis virus, which is sort of a lab rat or a lab pig. And so a great deal is known about mutational structures and selection structures for those viruses and a few others. I think that would be important. And then I had to come up with an acronym. So I proposed the Viral Evolution Evaluation Pipeline, VEEP, to integrate phenotypic and fitness predictions with evolutionary models, right? So try to build on what we know about other viruses to assimilate the information. And then the sales pitch, why will this be so great? Well, I think we'll be better able to quickly use genetic data and predict or forecast how worried we should be about different variants, and even just know what we should be studying quickly as new variants arise. Do they escape testing? Do they escape vaccination, and so forth? I think they'll make us much better able to couple genetic data with various other kinds of data. I've been ranting for a while that I think the clinical trial design for this pandemic was not done that well. I think looking at it from an evolutionary perspective, that is from the viral diversity perspective, might really help. And then the last point, which is what I already said, is we'll be able to quickly observe or respond to emerging genotypes. So I'll go to the chat in just a sec. This is the last slide. And so as much of challenges. So as I said, predicting the long-term evolutionary trajectory, rather important. Dealing with big data, in particular, this sort of validation and visualization of complex data is an important issue for all of us to be able to use it. And linking those data with models is key. Model integration and reproducibility, as we know, shows up everywhere. This issue that's certainly been on my mind a lot as we've worked through these questions is setting limits on predictability. I think that'll help us be more realistic about what we can do and realistic about what we can't do. As we know, there's always a great challenge in communicating evolutionary thinking to the general public. Here's evolution in your face. It's happening all the time. I think there's great opportunities here, and I hope we can use those to capitalize on this educational opportunity. And then, of course, this challenge, which I think all of the subgroups have, but I would say that's particularly true of this one, is that evolution affects all processes at all scales. And that therefore, if we believe the Dobzhansky quote at the beginning, and nothing in biology makes sense except in light of evolution, we should accept that as sort of being an underpinning of what we do throughout the subgroups. That's it. Okay, so that's all I had to say formally.
Fabulous. I guess, did you want to talk more about subgroup organization, or do you want to talk about the science?
I always want to talk about science. I don't ever hear about subgroup organization.
I guess I could ask a question because that's my privilege, I guess. Which is one of the problems I always have when I think about evolution, and you brought it up indirectly, and it may have been solved in your HCV, HIV examples, is that if I want to simulate evolution in a multi-scale model, I have to change parameters. And in general, what we do is we say we were going to do some Gaussian with such and such around the current parameter value. But there's no reason that a particular single nucleotide substitution in my viral genome should give me any kind of continuous change in any parameter whatsoever. And so, on the other hand, if you think about the effective evolution of a virus, you might expect that it doesn't matter so much which SNP it is. If the SNP causes a change of parameter of a particular type, then you should have a particular outcome. And so it seems to me there are two very different things, one of which you mentioned, which was going from sequence to phenotypic properties. And the other one was going from variation of some set of phenotypic properties to population evolution. Now, again, there's no reason that the variation of those phenotypic properties should be Gaussian or even continuous. It could be a total mess. But nevertheless, there are those two very different questions. And you mentioned, say, using molecular dynamics to do docking. And I can certainly imagine that if what I want to know is viral affinity to my receptor. But it's much harder to come up, although Kevin Janes a few months ago talked a little bit about this, about the possibility also of, say, doing a model of that type to look at interference with interferon response. But I thought maybe you could try to talk a little bit about what's been successful or unsuccessful in terms of actually looking at what the real distributions of those phenotypically related parameters are and how they map back onto the much easier to do sequence information. In this case, the single molecule things are easy. You can sequence very easily, but predicting the phenotypic parameters is hard.
Yeah, no, I agree with all that. I mean, yeah, I'm certainly not expert enough to answer all of those really interesting points, but certainly people have used the power of laboratory systems and directed mutagenesis to map out fitness landscapes, and they do not look like Mount Fuji, that's for sure, right? They've got these neutral networks, there's large flat areas, there's chasms in between them. What the consequences of that are for evolution, I would say, one is it is more unpredictable, and in particular, those fitness landscapes are context dependent. And so people can do them in the lab, but that may have very little to do with what happens when they have to deal with interferon. And second, I sort of see this as getting back to this predictability question, is how much can we imagine viral evolution looks like good old quantitative genetics, right? You get your cows, you breed them to make more milk, and they march up the mountain and make more milk. It really works because those are quantitative traits that, somehow underlying that are many genes of small effect. We know that's not true for viruses. They don't have many genes, right? And so we're looking at more Mendelian-like traits, fewer genes, a large effect. And at least in my view, I'd certainly be happy to be corrected. I think that makes the evolutionary process much more unpredictable because we're waiting for rare events, which are mutations, to take hold.
I mean, I guess somebody pointed out to me, actually, it was on the call a couple of weeks ago, that until very recently, SARS-CoV-2 still had a lot of ORFs. It wasn't even broken down into genes. And so that was a surprise that took so
long. So Fred, this was great. There's a wonderful summary of the key problems, especially the capabilities we need to develop. And not surprisingly, they're on the modeling side as well as on the data side. Can you say a little bit about the data side? What data we just can't find or can't generate, and which ones one could?
Yeah, once again, I only know my small corner of the world. I mean, the data that you always want to be able to do evolutionary modeling is linking genotype with phenotype, right? So we'd like lots of genotypes and lots of phenotypes. A problem that's arisen recently here in Utah, which, Wayne, is a good thing, is most of the testing is now antigen testing. So we don't even get genotypes, I should have put one of those trees, those huge trees that people have made of the entire SARS-CoV-2 lineage. But as I mentioned, very rarely can we link any of that genotypic information with phenotype. How sick did the person get? Young person, old person, male, female, whatever, ACE2 receptor, none of that stuff. And so to me, that's the biggest data gap. And I think there's almost no way around it unless one went, to Korea or somewhere that was willing to really design a study to do that really carefully. I don't think the somewhat haphazard way that we are collecting data now is going to have the power to make the genotype phenotype link.
But won't you also have to have the genotype for the human host as well?
That's even worse. Exactly. I'd be happy to have their age and the severity of the infection, but that's right. Layered on top of that is this huge variance generator of human genotype, which, as far as I know, we don't have any real smoking guns for genotype being that human genotype having that big an effect on the course of infection.
So generally, oftentimes the Veterans Administration tends to have the kind of data which are usually not so easy to collect.
This is an interesting point. So several years ago for my sabbatical, I signed up to work at the VA precisely for this reason, because they have a database of 30 million veterans. But the VA also has the world's most effective bureaucracy. And so I never actually managed to get my hands on the data. I didn't try that hard. But I don't actually know what's in there. I mean, even people in the group have trouble digging in and getting very detailed phenotypic data on the vets and usually the nasty drug-resistant bacteria that they've been studying. But it's a good idea, and I'll ask around and see whether anyone's actually been following COVID in the veteran population. And Jacob.
I have a question that's like struck me the minute you said something key that John Weiss keeps on telling me all the time. So it's like you said that viruses, well, you can't model what will happen through evolution. You said because it's too random, those are rare events. However, they do target all sorts of vulnerabilities in our system. Can we model the vulnerabilities we have and then rank them? So saying, okay, this is what we should look after. Instead of like modeling the viruses, model ourselves. Is it something that's useful?
I think it's a great idea. I mean, that's something that's fascinated for me for years. I honestly don't know if we really know enough about how the human cell works and how it's hijacked by viruses to do that. But yeah, in some ways that's my goal for understanding viruses. So I'd love to see some good examples of that.
Oh, I'm asking because John Weiss keeps repeating this idea to me.
Maybe he knows.
We're asking the expert now. You're the expert.
But he has no credibility in this field whatsoever.
What kind of experts did we hire?
Other questions?
I have I mean, it's sort of half question, half suggestion, but I wonder if, so, you know, if you imagine trying to do some kind of, well, Jacob was mentioning genetic algorithm. So imagine you do some kind of genetic algorithm that actually goes and tries to take a sequence and mutate it and derive in some very computationally intensive way something about phenotype, right? And try to map out the landscape like that. That would be, I mean, that doesn't sound feasible to me. That sounds like it would require far more computational resources than we have on the planet. Could we do this in vitro? I'm thinking of things like Colin Gett did with the genetic circuits. I don't know if any of you are familiar with that, but you know, this was an idea where, okay, this is in synthetic biology, trying to make synthetic genetic circuits that have some sort of a function, right? And the function is usually something terribly simple, like expressing a green protein. So taking a bunch of parts, a bunch of coding sequences and promoters and all these sorts of things, such that they assemble themselves together, mix them together in a big, in a big vat, well, vat or petri or whatever beaker or whatever they did, and then just chose the ones that had the right behavior and then sequenced them and said, oh, here's the answer. That kind of approach, maybe for, I mean, this also sounds terribly dangerous, right? Because if you were to, but if you wanted to say, well, okay, what sequences are reachable in small evolutionary steps that have higher binding affinity with some sort of a receptor, you might be able to find them in that sort of way by doing the computation actually in the wetware rather than in the computer.
Yeah, I mean, it sounds like a suicidal idea to do in your BSL-3 facility. But yeah, I mean, I certainly think if one were to take, so basically a stripped down system, like how does it get into a cell, right? So you have some cell line that expresses ACE2 and TMPRSS3 or whatever it's called, muck around with those things a little bit, maybe just how much of it there is, and then play around with the viral sequence in various ways to see how binding works. We would learn something useful, whether or not that would be a crucial component of the evolution of the virus in real life, we don't know. Ah, the airplane.
Does anybody recognize that
Oh,
image?
yes.
Of
What it
course.
means? So this
Yeah.
is from World War II. This is a study of where a particular aircraft came back to base with bullet holes in it.
Yep.
And the interpretation
that-
at the time was that we need to make armor everywhere you see one of that red dots. That's where we should be putting armor on the aircraft. And it took them about a year to figure out that that's ass backwards. That the places where you don't see any dots, that's where the plane crashed and didn't make it home. And so we have no data about bullet holes there. So if you put this in an evolutionary omics context, you ask the question, which nucleotides never change or rarely change? The reason they don't change is because if they change, it's a lethal mutant. So
Yeah.
all of these other places, you can do silent mutations and it makes actually no difference at all in the protein coding. You can also make conservative changes, which make relatively little changes, but there are a few places that you cannot tolerate changes. Anyway, just a thought, and it's a cool picture.
It's a great picture. No, I mean, that's certainly one of the bread and butter methods in evolutionary biology, right? You look for highly conserved regions, places that don't have bullet holes, and you're like, yeah, and so that's the game of, trying to find the universal influenza vaccine, right? You try to find a highly conserved region that you can target. And there's all kinds of, highly conserved and less conserved regions in SARS-CoV-2. But I think what the interesting point is that those rapidly evolving regions often are the ones that matter, because those are the antigenic regions where we see escape from immunity or escape from vaccine. So it's sort of interesting to think about the interplay between rapidly evolving as the stuff can actually conquer us, and slowly evolving, perhaps targets that we should be aiming at.
Yeah, the thought that most antibodies are to surface loops on proteins, and surface loops tend to be quite variant without really affecting the function of the protein. As long as they're hydrophilic, they work just fine. And so it's easy to mutate and avoid immune response without really changing much in terms of the structure and function of a protein.
Exactly. I mean, that's my vague understanding, and somebody else can clarify this for me, why a T cell response might be preferable to a B cell response, because T cells, particularly, ones that use class 2 MHC, are looking at proteins that got chopped up inside the cell. So you're looking at peptides that are not necessarily ones that are exposed in any way, and therefore you should have access to these highly conserved regions. I don't know if that's in fact true, but it certainly makes sense to me on a logical grounds.
Well, that's one of the tricky parts of protein folding, though, is that indeed the internal parts are not highly conserved. There's a great deal of flexibility of what amino acids pack into the hydrophobic core, and you can get a lot of variability in a hydrophobic core. It's almost as bad as the surface. The things that tend to be conserved are the active sites. They're much less tolerant of changes in amino acid sequence. But you can swap a phenylalanine for a tyrosine, for a tryptophan, for a proline, and it just doesn't make any difference when you're talking about a fold. The interior core of a protein is not a crystal, it's a melt. It's basically just hydrophobic goo.
That's right. But for those proteins that have a business end that's only expressed inside the cell as opposed to outside the cell, then T cells in principle have access to that, where antibodies don't.
Yeah.
Well, I think that the other, switching from the science to the bureaucracy, I think that was a beautiful case study in how to organize this kind of analysis. And if we could have a similar kind of workflow for doing that analysis approach for all of the other problems we have, it would be very, very valuable.
Thank you.
So, Reinhard, did you have anything you want to add to that?
No, I think this is great, and it's a real blueprint for what I envisioned the subgroups to do. And once that is done, then one can do exactly what Fred proposed, is to now link together to identify common problems, to identify common solutions, and work from there. I mean, it's sort of a big evolutionary process, and it will take some work. But I think if we can accomplish it, then we will have I think, made a great scientific roadmap, but also created a great connected community of researchers in the different parts.
Okay, we can come back to questions for Fred, I think, a little later. If John wants to present, we probably better let him do it now, because otherwise we'll run out of time in our allotted 90 minutes, 30 minutes, 60 minutes, sorry. And then, so John, do you have a material you want to present?
Yes, Jim, can you put up our slides? So, and this was advertised as presentation related to the IMAG meeting, but also to just a little bit of insight into what we're trying to do with the dissemination and outreach subgroup. So to speed it up a little bit, dissemination and outreach are a two-way kind of street. And so the first question we had is, what are we trying to disseminate? And then it's like, well, who are we trying to disseminate to? And then, well, what are the mechanisms that we currently have for dissemination? So what you're seeing is a very representative concept of the cube model that we use to try to think about the landscape in which we're trying to do dissemination and outreach. So I have a request for you, action item in this meeting. You see the list of audiences. That's a really small list. While I'm talking for the next couple minutes, if you guys would all go on the chat side and just short name as many audience identifiers as you can think of while you're half listening to the rest of this. Same way with what we call here the intent, or what is it we think that is so important that we need to disseminate in the first place. And so, the models themselves, knowledge from the models, are we trying to sell people on using something that they maybe are not comfortable with, or are we offering training? These kind of represent the models themselves. And so those two dimensions of our cubes need a lot of fleshing out. And what we have not done was try doing a survey monkey yet. So before we pester everybody with a survey monkey, if you would give some thought to this and throw them on the chat, and Bruce will capture those. Next slide.
John, you want specific names, or you want examples that are not on the list? What do you want?
Just something real simple with enough words to clarify what you mean. But I'll get back in touch with people who suggest things to follow up on maybe fleshing that out a little bit. This is just to capture a brainstorm list. If we had a flip chart and this was a brainstorming session, everybody'd be yelling out things and we'd be writing them down. Thank you. Next slide. So when we talk about dissemination and outreach, my experience in the modeling simulation consortium meetings is that we have created a dissemination work group there a long time ago. And kind of the central question was, the physicians and clinicians do not listen to us. Our models produce things and they don't listen. Well, okay, there's probably lots of audiences out there that don't listen. So the question is, how do we do a better job of dissemination? And all this attempts to represent is that one way you do that is you stop trying to push things out into a vacuum, but you take some effort to reach out and bring in people from the communities in which there are potential customers. So this is just a simple representation of that idea, and the idea that it's a chicken and the egg problem. if the physicians don't listen, but how many physicians have we invited to multi-scale modeling meetings to ask them what might be helpful to them that could come from multi-scale modeling if they had just a little bit idea what we were doing. Next slide. Okay, so currently we've done several things, kind of starting, it's a manpower problem, obviously, but these are some kind of generic topic areas that we've talked about developing papers on, doing brainstorm sessions on, focus teams, something to collect some data. One is simply in the area of how do you get information out? Well, the instructional technology and systems right now are becoming absolutely incredible. And guess what? They all depend on models. Well, why can't the model be one of your models that's producing an instructional program that's interactive, dynamic, animated, play with the variables, see what happens. The other is we have people very interested in how do we deal with the health disparities, and how does the concept of dissemination and outreach apply to minimizing adverse effect of not accounting for disparities that we understand are out there. Then within the dissemination outreach group, although there is a reproducibility and credibility subgroup, the key to dissemination is reproducibility and credibility. If the models are not seen to be creditable by the user, and the user doesn't know how to decide if a model's output is safe to use, then come on, you gotta start talking about how you improve that. That kind of goes along with the notion of just in general building trust in computational bioscience. This is a new world. You guys have been doing this for 15 years, but you do it among yourselves. And we meet at Bethesda for 2 days once a year, and we tell each other what we're doing. Well, that ain't exactly broad dissemination. So how do we start getting the message out to build trust so that people want to know about what we're doing? And then modeling the next pandemic begins now. This kind of goes along with the stuff that Fred was talking about. And the names here are people taking leads. And then the last area is communications between scientists and the media. If one of you had been approached by the New York Times to ask you to explain some model that they were going to do a report on in 8 hours tomorrow morning, going to be a headline banner, how do you do that in a way that something you said that you didn't really mean to say, taken out of context, is the 8-word headline on the newspaper that scientists said? How do we? So these are things that we're really interested in. And then what's just shown up that's really beginning to suck up time is this idea that we've been asked to host a replacement for the annual Bethesda live meeting, doing something virtual. So next slide. Jim. These are other kind of areas that we're working with. We want to try to develop some kind of personal relationship between computational models and the physicians. So one of our little side projects is I'm assembling, and we have an active member who's going to be coordinating a pool of retired practicing physicians that we recruit by convincing them that they don't want to totally give up learning, and that maybe if they would kind of join us and learn a little bit about what we're doing, we might actually pair a cardiac specialist who's retired with somebody building cardiac models and make friends out of them, mentoring friends, where the modeler can, where the physician can say, you know, what you're doing is really great. And if you could this, we could take that to the heart surgery conference and get a standing ovation. And it's not that you couldn't do it, we don't know. It's that you don't know that that's what they need. And similarly, to help you develop presentations to present outside of the modeling world, when you do want to take your models into the medical societies and associations. We also have you know, we recognized very early and talked about it here that we needed a list of who are the other organizations and stakeholders other than our working group that we at least freaking all know about. And we failed. We did not do a formal monkey survey on this either. But now that you've all put in your ideas for who the audiences are, if you want to type in a few names of associations in the chat list, that would be, you know, crowdsourcing some data to answer this question. We're also looking at the questions of how does the credibility question tie with what's really going on in industry and regulatory bodies. Bottom line, the FDA is now accepting some data from models and weighting the data to help reduce the time and cost of medical device clinical trials. The European Commission and the collection of European national health agencies know they need to do the same thing. Maybe multi-scale modeling concept has already halfway succeeded. You did it. Industry's using some of this stuff. The problem is industry has to convince the regulatory agencies that it's right. So that's just an area we're developing, and we're attracting some membership in the working group that represents those bodies. And then lastly, data management, data interoperability. I mean, yes, most of you heard the presentation that Catherine did. And that's because there are other areas other than biology that have been doing modeling and simulation for a long time. Believe it or not, they have. They're making money doing it. They're creating new stuff doing it. But some of them have solved the problem of how do they make their models talk to each other. So why don't we look at some of their solutions and see if they're adaptable? Next slide. This is just to kind of recognize, here's who we have as a membership list. We have weeded out people who checked the box that said they wanted to be members. And these are people who have been responsive. We exchange emails. The V people marked as visitors right now are all MDs. I mean, these are high-end, you know, Mayo Clinic retired superstars in their fields who said, yeah, maybe we would talk to some modeling kid and help them out. So I have to say Ruth is on maternity leave, and that has kept her from participating directly, but she communicates, and the blue people can participate regularly. Next slide. Now we want to talk a little bit about this meeting that we've been offered to host. Grace Pang offered and presented the opportunity for our outreach working group to maybe be the nominal host for a virtual meeting to replace the one that we did not have in March in Bethesda. So working with a lot of different people already one-on-one, kind of came up with this scheme that says, since we're virtual, we have an opportunity, a one-time opportunity to reach out into the world of people who are stakeholders in what you're doing and to bring them into our meeting. They're not going to travel to Bethesda for a 2-day meeting on the campus, but we might get them to come into a virtual conference setting. So essentially, we've proposed dividing the meeting into 4 separate kind of things. Okay, first of all, those of you who have been part of the IMAG and the consortium know that when we meet, the IMAG talks to us about their priorities. We talk to them about funding and things we want to do. The consortium has its own working groups. They have meeting time, the live meeting. They go to breakout rooms after we have scientific model presentations and stuff. So that still has to happen to some degree virtually. The big part is right over here, where it's the modeling on the left side, the modeling scientific content. And so what we're saying is we're kind of, something that Reinhard said to me really made this get a lot easier. The whole scientific part of papers is something you guys have done all your lives, all your professional lives. You know how to do that stuff. So right now what I need, heads up, tap, tap, pay attention. I need a couple people that will identify themselves as the co-chairs of the Scientific Content Committee. All that is coordinating and finding chairs for tracks. Tracks might be 1 or 2 sessions. We think of 1 session as a session, but if you want to have 6 presentations, you need 2 sessions. And so that would be a track. So now that you're done typing ideas about organizations, please, among you all, right of first refusal. If the subgroup that you chair has a title, which would be a track title, for which you could go find 3, 4, 5, call for papers. We'll talk about details if you're interested, to say, we want a track on multi-scale modeling applied to immunology, because Dr. Data has said she would be willing to co-chair an immunology track. So on the slides, she's got it. Jim, somehow I did something that I lost the slides. Everybody else seeing it? Anybody else see the slides?
I think he may have lost his internet connection.
Oh, bad news. I have a copy I can display because we only got a couple minutes unless you, and then those of you interested, please follow to the breakout time. But we'll give Jim just a second. Here he comes. He's back. And then the other major segment of this is over there in what we call the theme content. So unlike the typical conference where the keynote speaker talks about the theme and quotes it at the end of his speech, what we're saying is our theme, which is an outreach and inclusion theme, taking advantage of virtual, will be made up of sessions that include the topics kind of that we talked about our working group being interested in. So, Dr. Ferguson and Hernandez are working on a communications track that would be the journalist, maybe as a panel kind of thing, but then maybe even as a workshop the following week, where we go through role playing of how do you talk to a journalist that's trying to get you to say something they can make a headline out of. The whole credibility thing, we have co-chairs for that particular, these are like 1 or 2 panel sessions. We have industry and regulator people who have agreed that if they were asked, they would come do it. We have the data interoperability standards. This is just a way to single panels, discussions, just to open this stuff up for us to look at. And then lastly, because Grace has agreed that we could go into a second week, any of these things could turn into a workshop. If you have a workshop that you do now, and you want to post it, but have it on the IMAG agenda, this would be a way to say, we would like to do our workshop as part of the IMAG. And then, essentially you set up your own Zoom and it's scheduled for the following week. Okay, and then last slide. This is just kind of a representation of in a week how we can, whoops. This is like totally made up. Hour and a half sessions, because we want to kind of use what we do here. The hour presentation, and then there's always time to talk afterwards. That's the improvement over going to the movies. And so, plenary session, a keynote address for the science theme, which is virus, keynote address for outreach and inclusion. And then we start science tracks and session tracks. Nothing magic except that you can see we put in a lot of things. Notice the overlap is the next session track, which we would try to have not conflict with the topic here. It starts when the breakout starts in the one before, because we can put a lot more presentations in the time. And the idea is this is the one time when the presenter will be present. The presentation may even be pre-recorded, but this is the time that if you come at the scheduled time, then you have two-way communication after the event. Otherwise, you can click on any box anytime and watch the content. There's nobody to talk to, but you can watch it. And so you can catch up. It all works. Okay, that's the overview. I really am hoping that some of you that have subgroups will say you would like to talk about creating a track for your subgroup. And if you do, stick around in the breakout time. Jim?
Thank you very much. That was a great presentation. So we can open it up. Let's have a couple, if there are a couple of comments or questions initially for John, and then we could have a more general discussion. I don't know if we somehow could have describe, maybe we could evolve the optimal way to have a meeting. Except you'd think by now we've had enough experience with meetings that we would have evolved better ways, but okay. Go ahead, please.
The intent here was to share with you, this is more detail than anybody has seen, but you guys do your conferences. Tell me what's wrong too. We need to know what the problems are going to be. We know there's a lot, but we have some really good potential technical support to pull this off. That's impressive.
So, John, I have a comment on what you said about the FDA using models. It's a really important area that a number of people are focusing on. Helen Moore is one example. She has done quite a bit of work in that respect, what it takes to make models credible for a body like the FDA. It's also, I think, a place where modeling can have a huge impact. And the other thing is, I remember hearing a presentation by somebody from the FDA maybe a year or two ago saying that they would have a hard time considering models seriously that have not been published. And that is one big disadvantage that the industry has, because many industry models are not published.
Yeah, I was part of that FDA development. An organization was created by a nonprofit, by the Pew Charitable Trust, to provide neutral territory for the device industry and the FDI to get this problem worked out. Short story is FDI kind of looked at what NASA does, because after the Challenger, NASA got really serious about understanding models and their limitations. And so they kind of have the gold standard. FDI, the industry, looked at it. It's a lot of cost to the industry to be able to provide the required documentation. But if the FDA accepts it, it can cut millions of dollars off the cost of clinical trials for some parts. Nobody is trying to say the FDA is going to approve a device based on a model. But there are lots of parts of the model the FDA might otherwise test that they would test differently if they start with the model data. So Reinhard, you're right. This is big. It has now bled into, when you hear the FDA people talk about in silico trials, they are trying to look at the future of migrating from medical devices to pharmacology. And they ain't gonna get there without us. Yes.
Last week, we had somebody who nobody asked a question to, namely Natasha Go from Nova Discovery. That's a company that's now up to 50 scientists who design clinical trials, which are accepted by EMA.
Yes.
They really, I think that was a remarkably interesting talk. I'm sorry if I sounded grouchy. I shouldn't be grouchy. The Schiffer's talk was fantastic and people were excited by it. But I should have used breakout rooms to separate people so we could have two conversations.
Yeah.
But I think it would be great to have more talks like that.
Yep.
Because that really did show, and that wasn't its classic PBPK model. It wasn't a molecular dynamics model. They were combining in-host modeling with population level modeling, generating virtual populations, and really trying to model the clinical trial all the way at all scales. And to me, that was quite exciting to see that they were able to do predictive modeling that was able to show the effects, for example, of the lockdown on the clinical trials. I found it a fascinating talk. And the more people we could pull in that had that kind of experience and that kind of reach, I think it would be wonderful.
Yeah, we have. I've talked to all these people. I mean, I have people from the European health agencies that have said, yeah, I'll sit on a panel and tell you what the Europeans are working with. I got the guys from the FDA who said, yeah, we would love to do a panel on that for the modelers to help the modelers better understand what we're trying to do. So maybe they can do what they're doing in a more useful way. So the access to this, and I agree, I was fascinated by her thing, but it was like it was from another world or something. People didn't ask questions. So, okay, keep going. Lorenzo. Yeah, Lorenzo.
I have a question about, well, it's very general. We are talking for a while now about how can we show that the models are good, are valid, make sense, and they give us some prediction. The one thing I was thinking, essentially, is a mathematical model, if it's okay, well validated, nicely done, perhaps cannot predict particularly well what will happen, but it tells very often some good deal of what cannot happen at all. And could we find ways to actually highlight this bit? Because this is kind of dichotomic, no? This cannot happen, so stop thinking about it.
The reality of doing, that's why we need to be talking about this kind of stuff, but in the applied context. It's like, what are the trade-offs? What are the paybacks? How do we do this stuff? And I mean, I've asked questions about what is the model? Do you have to have a computer to have a model? And I'm going to argue, no. And you know what? Maybe you shouldn't be allowed to touch a computer until after you have a model. And then the question is the model make sense to lots of people? And if it does, then you get the mathematicians. And then the question becomes, is it verifiable? Does the math represent the model? And the FDA doesn't care about the math. If they'll, no, don't, I didn't say that. I don't care about the math. I'll get a mathematicians to work on that problem, and reproducibility becomes, it's the code good. But I ought to be able to hand my model in a book, a 1/2 inch thick, to 3 programmers who program the model in 3 different languages. And if we put the same input in the model, all 3 get the same answer. We don't do that. But if you look at what the FDA wants to know about the model, most of it would be in the book. They want to know what the variables are. They want to know what variables did you have on the whiteboard when you started that you crossed off. Because maybe one of those variables that you crossed off for what you were doing is the very variable that would make this important for what we're about to test. And so if I have a blood pressure model that doesn't have acceleration of the body in it, you know, how many of you ever saw a blood pressure model that accounts for body speed acceleration? It's great for the doctor's office. I can take any patient's data and predict their blood pressure when they sit in the doctor's chair. But if I put them on the front end of a spaceship and launch them, that blood pressure model is pretty freaking not creditable. So we need to start thinking that way. And anyway, I'm sorry, are any of you interested in taking on finding 3 or 4, 5 other papers? You can use a call for papers. Part of the idea is when we do our conferences, it's all us talking to each other. But here's a chance you can issue a real call for papers that says, I'm looking for papers that deal with immunology applicable to virus protection, whatever. And maybe we get 2 people who don't know that multi-scale modeling exists. The other thing that we've talked about is targeting small colleges and universities. You know, you guys from the big schools are not the only one in the world doing computational modeling. But we got colleges all over the world that have scientists doing something that can't even afford to apply for a grant. You know, well, you know, if we write the call for papers that says we are specifically interested in a couple papers from small institutions, you know, maybe we'll actually learn something. You know, more competition for NIH grants next year, but also more opportunity to collaborate on better grants. I'm talking too much. Other questions? Anybody want to talk about, by the way, Fred, I've talked to him about helping to just co-chair that whole segment on outreach and inclusion. By the way, the executive director for the European Virtual Physiological Human Institute is officially now a member of our virus working group and a member of our dissemination and outreach group. And she's a candidate for the outreach section with Fred because her outreach goes places that we can't even imagine. And she'll be doing a presentation in this meeting, in 2 or 3 weeks. So fascinating what's going on in Europe.
Great.
You're also quiet because I don't shut up. I don't.
I think there's so many issues that have been raised. No, there's so many issues that have been raised. I mean, people
Fred,
can also go back to talk to Fred about viral evolution and also about organization of subgroups, because there are many. I mean,
thank you for the time, and I was in no hurry to interrupt Fred's line of thinking because I really liked it.
So, I mean, one of the problems with this, I mean, problems maybe not be the right word. One of the things, the challenges, is that there's so many things that need to be done. And all of us have finite life expectancies and finite attention spans. And they're all interesting problems. The organizational problems are interesting. The standardization is interesting. The validation is interesting. And so how one breaks down these problems and finds a way that each of us can work productively on pieces of it, I think is still challenging.
I think one of the things I left out that's significant is this meeting is in November. We have 5.5 months. If you are willing to take on a track, I need a 1-page kind of proposal for what your track title would be and how many sessions of 2 to 3 each you're thinking you might need. Sessions don't have to just be papers. You can have sessions that are panels or stuff. So if you have some idea, some picture of a track that you would like to be the track chair of.
Do you think that if you're talking about calls for papers and that sort of thing, do you think that 5 months might be a pretty short window?
It's getting shorter.
Well, sorry, when you say paper, you really want there to be a publication, or do you just mean presentation?
Presentations. What we do here are fun. You know, I personally would like to see everybody tuned to present it in a way that we can put it on a public facing website and a 10th grade biology student could look at it and at least understand what the heck this modeling stuff is about. You know, how much detail can you put into 15 minutes that your colleagues can dissect the paper, and you get it out there and you're looking for people to discuss it with you afterwards and make connection and talk about collaboration around it. But otherwise, if we kept these things a lot simpler, this whole conference could be a dissemination tool. But any other questions for Fred? That's what I. Since you're all still here, we're talking about student papers by track. I mean, if in your track and you call, you say you welcome 10-minute student papers, and the concept we're playing with is it would be a recorded 10 minutes. And when people start your session at 11 o'clock, you don't sit around saying, let's wait a few minutes to see who logs on. The host of the session introduces the student and the 10-minute student presentation goes. And a heck of a lot more people interested because it's integrated into the track subject in that student's work are going to see it than if you set up an hour and try to do 5 10-minute student papers. And it's called outreach, integration, inclusion.
May I come back to something that you suggested before, which is slightly orthogonal to the conference, but it's a dissemination outreach group connecting to Reinhardt's desideratum of an inventory, which is you suggested using a prize model to get graduate students to contribute to an inventory of models. And the issue that comes up with that, I think it's a brilliant idea. The issue that comes up with that is that organizing the competition and administering it become critical.
I talked to Grace a little bit about competitions and some prize kind of stuff. And I'm going to go to William's comment. 5.5 months only seems like a long way off. And unless I have somebody stand up right now and say, I'll go find funding for prizes, we ain't likely to be funding prizes. You know, one hope is that, and in talking, I've got some really nice volunteer consulting support from some big conferencing organizations that have said, you know, we've been trying to do big conferences virtually. And what works is a system where each presenter has a time scheduled on Zoom with a video graphics technician. And you go on Zoom and you do your presentation, and it's 20 minutes long, and that's how long it is. And working with the professional, you back up, you can edit, you can send your slides to them, which means they can put it. You don't have any discussion of I'm waiting for my share screen, I didn't turn my microphone on. They edit the sound, they coach you to get your camera angle fixed if it's really bad, you know, try to adjust the light to get the best you can get from wherever you're recording, and they make a pretty professional recording. So when the session starts, you know, it's a recording. The fact that everybody came in at once means that after recording, there's a breakout group. But it's a high quality recording. And after the conference is over, that recording goes into the virus pandemic public website as a bunch of 15, 20-minute recordings of presentations for any of the ones that a normal person might say, whoa, I think I understand what they're doing. And so it's that format's pre-engineered. We're waiting to hear a little bit about some financing, but we have a low-cost academic institution that has the capability to do it. And panels, I mean, are really slick. The panel chair is on each one, but each panelist is kind of interviewed by the panel facilitator. And then they actually take the questions and they re-edit some of them and say, you know, we bounce from this question to this person, a different question to somebody else, and then come back to the first. I mean, and it's pretty, no wasted time.
Well, John, what you're describing is pretty similar to how this spring's AAAS meeting worked.
Yes.
And there were some really good aspects to it and some really not so good aspects to it. I gave a presentation in one of the symposia, and as I said, there were good things, not so good things. It did not feel like you were really interacting with a live audience at all. I mean, you record this thing, and then you have an interview with the moderator, and you have some comments in the chat.
Right. That's where our difference is, adding that half-hour breakout. And the other thing that I realized is if you take that schedule, that mock-up schedule, and you restart it at 10 o'clock at night, that the Australians and Japanese who join at the same time can talk to each other. If the presenter wants to wake up and be on there, that would be great. But the worst case is at least they can assemble in the breakout. Okay, but there's only one opportunity to do it with the breakout.
John, the Combine meeting did this last year with like a repeat every like several hours. It worked to some degree, but some things were lost, but it did work to the fact that some presenters did attend twice, and it was like a 24-hour rolling meeting. If you want to do it, it's okay, but you have to set up and make people aware that this is what's going on, which is kind of hard. And people will also split their attention. Think about it as well. But yeah.
But it's easy. It takes one night shift technician to be there, you know, to make sure the breakout rooms work at the night thing. And the other thing was the one big conference that had everything pre-recorded. You could look at any topic anytime in the whole week. There was no discussion opportunity, you know, but when you clicked on it, it's recorded. They were there.
So what you're also describing is something that's happening right now in the MIDAS annual meeting. I'm actually attending it. I skipped it to be here. But what they are doing is that they have the talks and then they break up into rooms. I can tell you that there's a little bit of disconnect. It's something that's not easy to manage to understand because people expect some sort of level of continuity also. But it's better than nothing, that's for sure.
We're experimenting. We had never lived in a world where we couldn't have live meetings. So we have learned a heck of a lot about some good things you can do. I mean, I have a list of things that are a problem with virtual meetings, and one of them is I got to bring my own snacks and wine. Well, can't solve that for you. You know, you're just going to have to bring your own wine and cheese if it's a, you know, if it's going to be part of the discussion, but we can't solve that. And you're right, Reinhard, giving a presentation to a blank wall, but getting your information out nice, clean, and crisp, and being perfectly comfortable that anybody in the world could watch that any day and you would be proud of it. Maybe nobody watches you.
So John, one thing that's important, usually there is usually some sort of like a mechanism that organizes everything, like a website or something like this. Did you select one already?
I got chats were covering up my closed caption.
You need to select a website to organize this.
This is a group that does it all. We don't have to screw with it.
Okay.
I mean, we may not be able to do that, but we can't get to finding out until I have at least 4 or 5 track themes with people who had said that they'll do them. I mean, there's no point in trying to tie up other volunteers' time if I can't very quickly, for Grace, produce a list of what the topics were. Originally, she did not ask. You know, she wanted the construct of the meeting, but it's 4:30, and but now she's saying it would help to know what the topics were. Well,
until when do you need all of this done? Because the faster it is, then it'll work.
We'll see.
Okay.
Yeah, I've gone to sea in rough water before.
Okay.
You know, I just need some of you willing to get out there and bounce around with us. Okay.
So we're out of time.
Yes.
I'm impressed that you were able to keep so many people on so long to discuss this topic, which I think is an important one. I'm not being critical of it at all. I think it shows that people are excited by this idea and want to see it happen.